welcome to section 5F. In this last section, we explore AMU-C surveillance in animal health. The goal and objectives of AMU-C surveillance in animal health are similar to human health, and the definitions of AMU and AMC similar. However, in low and middle income countries, collection of AMU data is difficult as the records are often poor. Therefore, the main surveillance method in the initial phase is AMC in quantity. This slide summarizes the goal and objectives of AMU-C surveillance. The goal of AMU-C surveillance is to generate knowledge and evidence needed to promote optimal antibiotic use in the animal subsector and for the effective containment of antimicrobial resistance. Objectives: monitor consumption trends and benchmarking for optimization of antimicrobial consumption, facilitates identification of trends and usage profiles, permits establishment of time-bound consumption targets, monitors progress toward achieving targets, guides policy and targeted interventions, optimizing antimicrobial use, comparison of antimicrobial consumption data across countries, species, farms, and with human consumption, builds a platform for studying the association between antimicrobial consumption and AMR surveillance data within the animal health sector and externally with other sectors. Antibiotic use. This slide summarizes the definitions of AMU and AMC in animal health. Antimicrobial use data refers to estimates derived from animal level data. These data may allow disaggregation based on the sick animal characteristics, species, gender, age, or indication for which the medicine is being used. This will facilitate assessment of clinical practice against agreed protocols and treatment guidelines. Antibiotic consumption. Consumption data refers to aggregated data where there is no information available on the animals that are receiving the medicines or why the antimicrobials are being used. Provides a platform for studying the relationship between AMR and AMU-C within animal health and among other sectors. Sources of data will vary from country to country. However, in many countries, this may include customs services, import and export data, manufacturing and sales data, data from veterinary medicinal product registration authorities, wholesalers, retailers, pharmacists, veterinarians, feed stores, feed mills, and pharmaceutical industry associations. Calculate and monitor population correction unit, aggregate antibiotic consumption measure for livestock. This slide demonstrates a phased implementation for AMC surveillance. Phase 1: assess organization, leadership, and management structures in animal health sector. Phase 2: determine sources of drugs and medicines for the animal health sector and map distribution and supply chain pathways. Phase 3: define surveillance indicators, methods or protocols and data collection tools and collect data from pilot sites, farm level, districts, national level. Refer to OIE and FAO guidelines. Phase 4: at completion of pilot, establish a system for data collection, analysis and reporting at relevant levels. Report data to NCC or TWG, WHO, FAO and OIE. This slide shows the initial phases of an AMU-C surveillance program in animal health. AMC data is collected from National Drug Authority, NDA, and this can be annual estimates of volumes of antimicrobials or antimicrobial class imported or locally manufactured. This is expressed in kilograms per population correction unit. Monthly estimates of volumes distributed based on sales data from distributors. quarterly estimates of volumes of antimicrobials used on the farm. Please note that under AMU, there may be qualitative description of practices that are associated with use of antimicrobials on the farm during AMR surveillance data collection.